Hello students, in previous part we have studied about different types of lipid. In this section we will discuss on nucleotide, nucleoside and DNA. What is a nucleotide? Nucleotides are microbiomolecules. These are microbiomolecules. But when many microbiomolecules means nucleotide combined to form a huge chain that becomes the higher in weight and that change into macrobiomolecules. Firstly, we will discuss or we will differentiate between the nucleotide and nucleoside. Nucleotide. As we know that nucleotide is microbiomolecules, it is lesser in weight. Nucleotide has different composition, pentose sugar, nitrogen base, or phosphoric acid. Phosphoric acid, mostly in phosphate form. Phosphate form. These are the main components of nucleotide: pentose sugar, nitrogen base, and phosphoric acid. Pentose sugar. Pentose sugar is a ring of 1 oxygen and 4 carbon atoms. Pentose sugar, actually pentose sugar is also two types: ribose sugar and deoxyribose sugar. Ribose sugar is in the part of RNA and deoxyribose is the DNA. What is the difference between ribose and deoxyribose? We will discuss later. Firstly, what is the pentose sugar? Pentose sugar is a ring in which 1 oxygen molecule bonded with 4 carbon atoms and 5th carbon remains in the form of free arm. This is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this is the carbon number 5. It is the structure of pentose sugar. Whether it is ribose sugar or deoxyribose, both have this one like structures. But the major difference between ribose and deoxyribose we will discuss later. Second one is nitrogen base. Nitrogen bases are also two types. There are two types of nitrogen bases: purine and pyrimidine. Purine is also two types: adenine and guanine. Adenine represented by A, guanine with G. Pyrimidine is of three types: thymine, uracil, or cytosine. Pyrimidine is of three types, but when we make the RNA or DNA, only two are used. If the RNA, then thymine and uracil is used, and when it is DNA, in place of uracil, thymine will come also. This is the nitrogen base. Phosphoric acid. Phosphoric acid, which is the P molecules, phosphate molecule that combined with the 5th carbon of pentose sugar ring. How is this structure can be produced? Firstly, we will draw the structure of pentose sugar. Pentose sugar is a ring of 5 carbon molecule, this one, in which 4 carbon attached with 1 oxygen and form a ring, and 5th carbon remains here in the form of free arm. Carbon number 3, 4 and 5th one. At carbon number 1, purine and pyrimidine, the nitrogen base. Nitrogen base is attached as carbon number 1 and carbon number 5th is attached with the phosphoric acid. Here it forms the ester bond. What is the ester bond? The bond which is formed eliminated of one water molecule between the carboxylic and hydroxylic group. That is called, here bond is formed between the, this is the OH contain. We will discuss OH, and this phosphoric acid is attached with this one. This is called ester bond. How is the difference between ribose and deoxyribose pentose sugar? This is the only pentose sugar. But when we differentiate between ribose and deoxyribose sugar, we will draw the one more structures. This one is also pentose sugar, carbon number 1, 2, 3, 4, and this carbon number 5 is remains in free arm. Same structure. In ribose and deoxyribose, this is the major difference. At carbon number 2, here is OH and H, and carbon number 3, here is H and OH. Same structure is here. This is the carbon number 1, 2, 3, 4, and 5th one here. At carbon number 2 in a deoxyribose, oxygen molecule is reduced, so it is called deoxy. And carbon number 3 remains such like this one, H and OH. But there is a difference between at the carbon number 2nd position. If carbon at 2nd position contains H and OH, that is the ribose sugar. It is a part of RNA. And if at carbon number 2 it reduces one oxygen molecule and forms H and S, this is the DNA. Because in this structure one molecule of oxygen is reduced. So this is the major difference. Second one is RNA has more functional group. It has 2 hydroxyl group at carbon number 1 and 2. But DNA contains only one. At carbon number 2 there is no hydroxyl group. So due to the less number of functional group, DNA is stable because it is not so active. RNA contains more hydroxyl group, more functional group, and we know that the compound which has more functional group, that is more active. If it is more active, then that not remains stable. Due to this stability, DNA is known as genetic material. This is the major difference. First one, purine and pyrimidine both are nitrogen base. Purine is of 2 types and pyrimidine of 3 types. But when we draw the structure of RNA DNA, only 2 pyrimidines are used. This is the first difference. Second difference between the purine and pyrimidine is purine is dicyclic. It forms dicyclic. It is dicyclic. And pyrimidine is monocyclic. Third difference is in purine there are 4 nitrogen atoms. 4 nitrogen atoms. And they attach at carbon number 1, 3, 5 and 7 and 9 position. They attach the nitrogen bond, or nitrogen atoms are attached at carbon number 1, 3, 7 or 9 position. But in monocyclic or pyrimidine, it contains only 2 nitrogen atoms, and they attach at carbon number 1 and 3 position. This is the major difference between purine and pyrimidine. Purine always bonded with the pyrimidine. Purine adenine always bonded with the thymidine with a 2 hydrogen bond. This is not the sign of is equal to, it is the 2 hydrogen bond. Adenine always, if the DNA, then adenine combined with thymine with a 2 hydrogen bond, and guanine with cytosine with a triple hydrogen bond. If we draw the structure of DNA, this will remain same. And if we draw the structure of RNA, then adenine bonded with 2 hydrogen bond with uracil, and guanine with a triple hydrogen bond with cytosine. This the, if we draw the structure of RNA, this will remain same. So RNA and DNA, two major group, two major differences. At carbon number 2, RNA hydroxyl group, but in DNA there is no hydroxyl group. Second one, adenine always bind with the uracil with a 2 hydrogen bond in RNA, but in DNA adenine bonded with the thymine in place of uracil. 2 hydrogen bond. Guanine and cytosine always remain same. Clear? Now how this structure can be bonded with each other? At carbon number 1, nitrogen base are attached. Here is nitrogen base, for example here is adenine is attached. This bond between the carbon number 1 and nitrogen base, this bond is called glycosidic bond. The bond which is formed at carbon number 1 between the nitrogen base, it is not only A, maybe any nitrogen base. Carbon number 1 when combined with the nitrogen base, a bond is formed. That bond is called glycosidic bond. And phosphate always attached with the 5th carbon, that forms the ester bond. We know that ester bond, when carboxylic acid combined with the hydroxyl group with the illuminate of one water molecule, that is called ester. But mostly it forms diester. How is the diester? If this phosphate is again here attached with the hydroxyl group, one is hydroxyl, other is there are 2 hydroxyl group, so it is called phosphodiester bond. Actually, if only it combines one side from hydroxyl, that is called phosphoester bond. Phosphoester bond. One it combines, phosphate combined with one hydroxyl group, that is, it is called phosphoester bond. But when phosphate attached with the both side hydroxyl group, that is called phosphodiester bond. Same in DNA. Here is nitrogen bond, nitrogen base is attached with the carbon number 1, this is called glycosidic bond. At carbon number 5th, here phosphate is attached, and that was the phosphoester bond. If both sides phosphate is attached, then that is called phosphodiester bond. This is the structure of DNA and RNA. How is the major difference between the nucleotide and a nucleoside? Then what is nucleoside? Nucleoside is also micro biomolecules. Nucleoside. Nucleoside contains only pentose sugar and nitrogen base. There is no phosphate molecule is present in this. Nucleoside doesn't contain phosphate. If it contains phosphate, then this nucleoside change into nucleotide. This is the major difference between the nucleotide and nucleoside. So we can say that nucleoside is less in weight than the nucleotides because it doesn't contain any phosphate molecules. How is the DNA and RNA? How the nitrogen base are recalled in RNA and DNA? As we know that in nucleotide and nucleoside, again nucleoside, it contains only pentose sugar and nitrogen base. So how we recall the nitrogen base in a nucleotide and a nucleoside? Nucleotide or nucleoside. As we know that in nucleoside only pentose sugar plus nitrogen base. There are 5 nitrogen base, means purine and pyrimidine. There are 2 purine and 3 pyrimidine. If it contains pentose sugar and adenine, then that is called adenosine. If pentose sugar plus G, then that is called guanosine. Guanosine and adenosine. But when pentose sugar combined with pyrimidine, such like as thymidine, then this is called thymidine. and pentose sugar with uracil, that is called uridine, and pentose sugar with cytosine, that is called cytidine. We recall these nitrogen base in nucleoside and nucleotide. These are the nucleoside. In nucleotide, we know that pentose sugar with phosphate. So we can say that pentose sugar plus phosphate plus nitrogen base A, then that is called adenylate acid. Adenylate acid. Why it is called acid? Because it contains phosphate, and phosphate diester bond means phosphoric acid. Due to presence of phosphoric acid, it is slightly acidic in nature. Slightly acidic. Which one? Nucleotide, because it contains phosphoric acid. But nucleotide sides contain only pentose sugar and nitrogen base. There is no acid, so it is slightly basic. So on the basis of phosphoric acid, it is slightly acidic, adenylate, and pentose sugar plus phosphate plus guanine, then that is called guanylic acid. If it contains pentose sugar plus phosphate plus thymine, then that is called thymidylate acid. And if contain pentose sugar plus phosphate plus cytosine, that is called cytidylate acid. If it contains pentose sugar plus phosphate plus uracil, that is called uridylic acid. So this is the major difference between the nucleotide and nucleoside. In next we will discuss on DNA or RNA, because RNA and DNA both are nucleic acid. Next one is DNA. What is DNA? Nucleic acid. There are two nucleic acid. One is DNA and other one is RNA. First we discuss on DNA. DNA was discovered by Frederick Miescher in 1869. But the structure of DNA was proposed by J.D. Watson and Crick. The double helix structure of DNA was proposed by J.D. Watson or Crick in 1953. What are the features of double helix structures? Features of double helix structures. Features of DNA structure. First one, DNA is double helix, means it contains double strand. DNA is a double stranded. It means it contains two strands. It is a, yeah, DNA is a double helix. Helix means a stair-like, a stair which contains two long arms that make the side bones or backbones, and there are small step-like bones that make the nucleotide bones or nitrogen base. Sorry, double helix structure. As we know that DNA is double-stranded, it means contains two strands, and polarity of the both the strand is opposite. Both the strand has opposite polarity. How is the opposite polarity? DNA is a double-stranded and both strand has opposite polarity. If one strand contains 3' to 5' polarity, suppose this is the DNA double helix structure, double-stranded. If one strand contains polarity 3' to 5', then other will show opposite polarity. It means it contains 5' to 3' structure. How this structure we will discuss later. These are the features. Next, both strands run anti-parallel to each other. Both the strands run anti-parallel to each other. How? Because we know that both the strands has opposite polarity. Opposite polarity, it means that both the strands are anti-parallel to each other. The DNA in one turn contains 10 nucleotide. If it contains 10, then we can say that there is DNA B, because on the basis of nucleotides, DNAs are 5 types: DNA A, DNA B, DNA C, DNA D, and DNA Z. If it contains mostly, the DNA contained in one turn contains 10 nucleotide, or we can say that, 10 base pair. 10 base pair, nitrogen base pair. That is called a pitch. So, distance of a pitch is 3.4 nanometer, or we can say that 34 angstrom. So, the distance of one pitch, one turn that completes in 360-degree angle, one pitch or one turn is 3.4 nanometer or 34 angstrom. Firstly, draw the structure of DNA. As you know, DNA is a double helix structure. Suppose the single strand, and this is the second double-stranded structure. One strand come back here and it forms this one like. Because from here it comes down and here it becomes upper side. this one like structures. It means DNA is a double strand, one strand here, other is here. It always, mostly DNA parts, it coiled clockwise or right-handed. Suppose we have a stair and which have both side railing, then it will turn this one like clockwise. That DNA is the double stranded structure, and one polarity of one pole is if 3 prime here, then it is the next one is 5 prime. Second one is 5 prime. This contains 3 prime. So DNA is a double stranded structure. This is the DNA double stranded structure. Second one is both the strand has anti-polarity. It means both the strands runs anti-parallel to each other. If one strand contains 3 to 5 prime polarity, then other will 5 prime to 3 prime. Both the strand always turns right-handed. We can say that both strand turn right-handed, or we can say that clockwise to each other. These are the backbone. These are two strand, and two strand makes the skeletal or backbone. The backbone or skeleton of DNA is made up of pentose sugar and phosphoric acid. These are the two strands, and both strands are known as backbone. So these are made up of pentose sugar and phosphoric acid, phosphate. And both strand has depression. There are two depression. This is the shallow depression. This is the deeper depression. This is the shallow depression. Shallow depression, or we can say shallow grooves. And this one is deeper grooves. The distance between two consecutive shallow to shallow, it means from here to here, or deeper to deeper. The distance between two consecutive shallow to shallow grooves or deeper to deeper grooves is known as a turn. Or we know that turn is known as also a pitch. It means from here shallow grooves to shallow grooves. This distance is known as a pitch or a turn. This turns only at a 360 angle. And one turns contain, here is nitrogen base. Both the backbone strands are attached with the nitrogen base. This is the N to nitrogen base. One turn contains 10 nitrogen base. From shallow to shallow grooves or deeper to deeper grooves, that is called a pitch or a turn, and turns at a 360 angle, which contain 10 nitrogen base pair. The distance from shallow to shallow, deeper to deeper groove, a pitch, is 3.4 nanometer or 34 angstrom. So we can say that if it contains 10 base pair, the distance between two consecutive, total distance here 3.4 nanometer, or we can say that 34 angstrom, one pitch, and it contains 10 nitrogen base pair. So distance between two consecutive base pair is 0.34 nanometer or 3.4 angstrom, because it contains 10 base pair. So we divided 3.4 by 10. The distance between two consecutive base pair is 3.34 nanometer or 34 angstrom. 3.4 angstrom. As we know that DNA is double stranded, both strands run parallel to each other. So if we draw a rough line through the center, this center, it means this is the in radius, or this is the spiral, and so the diameter, it means the distance from here, this one is the diameter between the two base pair is 20 angstrom, or we can say that 2.0 nanometer. This one like structures may present in DNA. How both the strands are present? We will draw the rough structure of both strands, how these are attached with each other. As we know that DNA is a pentose sugar, and pentose sugar form a ring of 4 carbon molecules, atoms, and 1 oxygen. Suppose this is the pentose sugar. Carbon number 1, 2, 3, 4, 5 is here. Here is H and H. Here is H and OH. Because this is the structure of DNA. One more molecule of nucleotide is this one, like carbon number 1, 2, 3, 4, and 5th is here. Here is H and OH. This is the carbon number 3. This bond, it contains phosphoric acid. Here is OH. If this phosphoric acid attaches here, this forms the phosphoester bond. But when it attaches, this one is also, then there is 2 phosphoric acid. So it forms the phosphodiester bond. Phosphodiester bond. This is the 5th position. So if we draw the single line, single strand of this one, like here is the 5th is here free, and here is 3. So one strand is 5 prime to 3 prime. And second strand, how the second strand is produced? At carbon number 1 we have studied carbon number 1 nitrogen base is attached. Here is also nitrogen base, and the base and the bond which is formed at carbon number 1 with nitrogen base, that is called glycosidic bond. Glycosidic bond. How these are attached? If we draw some another in the another way, this one, like this is the carbon number 1, this one carbon 2, 3, 4, and this one is 5th here. It will contains OH. OH. Carbon number 1, it contains nitrogen base. Here is the dependent which type of nitrogen base is present. If here is adenine, purine is present, then it must be thymine present here with attached with a 2 hydrogen bond. But if it is RNA, thymine in place of RNA, it comes UAC. Another bond, this one, this is the carbon number 1, 2, 3, 4, and this one is 5 here. 4, 3, it may contains H and OH. It is H and OH. And 5th, phosphoric acid is attached. Here is OH. This bond is known as phosphodiester bond. If we draw this one, a single line, it has polarity 3 to 5. This one was 5 prime to 3 prime. This is 3 to 5. So we can say that. And if here is attached with, for example, here is guanine. Guanine is attached with cytosine with a triple hydrogen bond. These bonds, so we can say the backbones are contained with the hydrogen bond double or triple. This is the structure of DNA. Now, how many types of DNA? There are 5 types of DNA. On the basis of presentation of base pair as well as turn in coil. Types of DNA. There are 5 types of DNA. DNA A, DNA B, DNA C, DNA D, and DNA Z. There are 5 types of DNA. These 5 types of DNA are basically categorized. First one is on the basis of bases of base pair. Nitrogen base pair. DNA A contains 11 base pair. DNA base B contains 10 base pair. DNA C 9.33 base pair. DNA D 8 base pair and DNA Z is 12 base pair. We have discussed all DNA B because we have discussed there are 10 base pair. So the DNA which contain 10 base pair, that is DNA B. Second one, the direction of coiling. We know that both the turns, both the strand are run anti-parallel in coiling in turns to each other. The direction of coiling, DNA A, B and CD both are always turning right-handed. But DNA Z is an exceptional case. It turns in left-handed. These are the 5 different types of DNA. In next section, we will discuss on different types of RNA, RNA, mRNA, tRNA and rRNA.